Today's topic is large glands of digestive system. These glands include 3 pairs of major salivary glands, liver, and pancreas. Also, we will discuss the structure of gallbladder, the sac where the secretion of liver is stored, the bile. All the glands are the parenchymal organs. It means that they have the parenchyma, the mean tissue, and the stroma, the supportive tissue. The supportive tissue is, in all of these glands, loose fibrous connective tissue with blood vessels and nerves, and the parenchyma is the epithelial tissue. It includes the cells of secretory portions and the cells of excretory ducts. And the embryonic source of development of the parenchyma of salivary glands is ectoderm. The embryonic source of development of parenchyma of liver and pancreas is endoderm. So, in the glands which develop from ectoderm, in secretory portions we will have the myoepithelial cells. In glands which are produced from endoderm, there are no myoepithelial cells. And the excretory ducts in the ectodermal glands usually have 2 or more layers of cells. In glands which derive from endoderm, all excretory ducts are always lined with simple epithelium, one-layered. Okay, we will start with major salivary glands, which include 2 pairs of parotid salivary glands, 2 pairs of sublingual, and 2 pairs of submandibular salivary glands. Their secretory portions are located outside of the oral cavity, and the ducts lead to the oral cavity. Functions of salivary glands are secretory function. It means that they produce saliva, which includes proteins such as amylase, maltase, and antibacterial substances, and mucus. Excretory function, it means that through the salivary glands, some toxins and wastes can go out from our body. This function increases when the major excretory systems fail, such as kidneys. So in kidneys disease, the salivary glands will excrete more toxins and wastes. Endocrine function. The salivary glands also produce some hormones and biologically active substances. And regulation of water-salt balance. It means that they produce some ions in structure of saliva, and they also regulate the pH level. And the regulation of water-salt balance is more important for the body also in the case of kidney failure. The structure of salivary glands. All salivary glands include secretory portions and excretory ducts. The secretory portions can be of 3 types. Serose secretory portions. They include cells serocytes and myoepithelial cells, which lie upon the same basement membrane. The serocytes are the protein-producing cells. They produce enzymes amylase and maltase, the enzymes for digestion of sugars, antibacterial proteins, lysozymes and lactoferrin, and secretory component, which binds to immunoglobulin A and helps to transduct, to transport this immunoglobulin produced by plasma cells outside of secretory portions in surrounding loose connective tissue. So these plasma cells would produce immunoglobulin A. It then comes into serocyte. Serocyte produces secretory component which binds to immunoglobulin A and protects this immunoglobulin from intracellular proteinases, the enzymes that would break this immunoglobulin. So the immunoglobulin is protected, and it is transported through the cytoplasm of the serocyte and is released with the saliva into the lumen of secretory portion and then through the ducts. So our saliva also contains immunoglobulin A, produced by plasma cell. The myoepithelial cells are present in all secretory portions and also in the ducts, intercalated and striated ducts. Two first kinds of ducts. Myoepithelial cells have long processes which hug the excretory ducts and secretory portions from outside. In those processes, they have the contractile actin and myosin microfilaments, and when the cells receive signal from for example, such as the hormone oxytocin, they can contract, and this causes the squeeze of secretory portion and release of secretory granules from the cells, serocytes and mucocytes, into the lumen of the secretory portion and then into the ducts. Mucous secretory portions, they contain mucocytes and myoepithelial cells. Mucocytes have transparent cytoplasm because it is filled with the granules of mucin, which is not stained neither with hematoxylin nor with eosin. These granules fill all the apical portion of the cell, and they squeeze and move the nucleus, which becomes flattened, to the basal portion of the cell. Unlike the mucocyte, the serocytes have basophilic cytoplasm. It is basophilic because of rough endoplasmic reticulum needed to produce proteins, and the nucleus is round. Myoepithelial cells are present in both cases. And also we can have the mixed type of secretory portion, which contains both secretory cell types, mucocytes and serocytes at the periphery. Mucocytes produce mucus, which is a jelly-like substance which can absorb a lot of water. Major component is the mucin, which is glycoprotein, so the organelles which can be found in the mucocytes include rough endoplasmic reticulum and Golgi complex, because the glycoprotein molecule contains the protein component and glucose component, sugar component. Next, after the secretory portions, the excretory ducts start, and first two types are intercalated ducts and these striated ducts. Intercalated ducts are composed of one layer of cuboidal cell of the lining epithelium and myoepithelial cells. This is how it looks like on the cross-section. Small cuboidal cells with small amount of cytoplasm. They look very small on histological sections. Also in these areas, in intercalated ducts, there are stem cells for both secretory portions and excretory ducts. Then, after intercalated ducts, the striated ducts go. They are composed of columnar epithelial cells and myoepithelial cells. They are called striated because they have this striation in the basal portion of the cell. The striation is caused by the presence of a lot of mitochondria, which serve for the functions of these ducts. It will be discussed later. All major salivary glands are compound and branched. Compound means that their excretory ducts are branching, and second word, branched, means that several secretory portions lead to one duct. But the difference is in the shape of the secretory portion. The parotid gland are compound branched alveolar. It means that they have only alveolar type of secretory portions. Submandibular are compound branched tubular alveolar, And sublingual are also compound bridge tubular alveolar. It means that they have both alveolar, round secretory portions, and tubular, elongated secretory portions. The shape of secretory portion depends on the type of producing cells. So in parotid, we have only serocytes, which form round alveolar secretory portion. And the mucocytes form tubular secretory portions, or mixed secretory portions will be alveolar tubular. In parotid gland, we have only serous secretory portions. Here they are. They are round and the cells are basophilic. Submandibular gland has mixed mucoserous secretory portions, which include both mucocytes and serocytes at the periphery, and also serous secretory portions. In sublingual gland, we have mostly mucous secretory portions, but also some amount of mixed ones. This is how these three glands look like on histological sections. In parotid gland, we have the serocytes, which are stained equally, and these secretory portions have the basophilic cells and not very well seen lumen of the secretory portion. In submandibular gland, we have mostly mixed secretory portions. In sublingual gland, we have mostly mucous secretory portions. So the salivary glands differ by secretory portions, but they have common principle of excretory duct system organization. This is very important. You should learn these four types of excretory ducts. So the first duct, which comes just after the secretory portion, is the smaller one, which is called intercalated. It is composed of small cuboidal cells and myoepithelial cells. Cuboidal epithelium and myoepithelial cells, and these ducts contain stem cells for both secretory portion and ducts. Then goes striated duct. After the intercalated one, striated duct contains columnar epithelial cells, which function is to pump sodium and water from saliva through the cell outside to surrounding loose connective tissue and in blood, and to pump potassium and bicarbonate ions into saliva. It means that in the membrane of these cells of striated duct, there are a lot of ion pumps or ATPases, which use the energy of ATP to pump some ions into the saliva and some ions from saliva to the blood. And these ion pumps are located inside the membrane, and they need the mitochondria to produce energy. So they have this striation filled with mitochondria. This is why these ducts are called striated ducts. They also are surrounded with myoepithelial cells. After striated ducts go intralobular ducts. They are two-layered, double-layered cuboidal epithelium and no myoepithelial cells. They are called intralobular because they are located inside the lobules of the glands. The cells of these ducts just convey saliva. They do not produce anything and they do not have any other function. And then finally go interlobular ducts. They are the most large. They have 2 or 3 or more layers of epithelium, so the epithelium there will be striated, and they also just convey saliva. The largest one duct will be the major duct, which leads to the oral cavity. Here is the function of the striated duct. So this is how it looks like on light microscopy. The cells are oxyphilic because they have a lot of mitochondria, which are located around and mainly below the nucleus and form the structure which is seen on light microscopy as basal striations. These lines in the basal part of the cell. If we look at the cells on electron microscopy, we will see that the cells have two components: folds of membrane, invaginations of membrane in the basal part of the cell, and a lot of mitochondria between these folds of membrane. Folds of membrane increase the cell surface area to increase the amount of integral proteins, which perform the function of transport of ions and water across the cell membrane. So these integral proteins are channels and pumps, ion pumps, such as sodium potassium ATPase. So there are a lot of such pumps incorporated into this membrane, which forms the folds here. And ATPases need the energy of ATP. So mitochondria produce ATP. This is how such cell looks like on electron microscopy picture. This is the border of the cell, because here we see the intercellular junctions. Here, this is the basement membrane. And we see here a lot of folds of membrane from the basal portion of the cell. And between these folds, we have a lot of mitochondria. Mitochondria make oxyphilic staining of such cells. So the function is active transport of ions into or outside of the cell. And exactly for striated ducts, they pump potassium and bicarbonate ions into the saliva from blood and pump the opposite way the sodium and water from saliva. So the function of this is to add some bicarbonate ions into saliva. Bicarbonate ions would neutralize acids, so they serve for the correct pH balance in the oral cavity. And the water is pumped out from saliva to make the saliva more concentrated. This is how the section of the submandibular gland looks like in the drawing. You have this picture in your album. You should colorize the picture using the correct colors. So in submandibular gland, we have two kinds of secretory portions. Serous ones, they contain only basophilically stained serocytes and myoepithelial cells. And mixed secretory portions, they contain non-stained transparent mucocytes with flattened nuclei and basophilic serocytes and myoepithelial cells. So these are two kinds of secretory portions. And now about the ducts. The smallest one duct is the intercalated duct. It looks like the small group of small nuclei of the cells. This is it here. Here is also intercalated duct, and here intercalated duct. Intercalated ducts then pass into striated ducts. So here we have the striated duct. It is composed of elongated columnar cells which are oxyphilic because of a lot of mitochondria. So these oxyphilic structures are striated ducts. They also are surrounded with myoepithelial cells as well as the intercalated ones. And here we have large layer of loose fibrous connective tissue which separates the tissue of the gland into lobules. So this will be interlobular connective tissue. And here we will have the interlobular excretory duct. It has three or more layers of cuboidal cells in its wall, so it is lined with stratified epithelium, and no myoepithelial cells are present. So this is the interlobular excretory duct. We should also have interlobular one, intralobular one, which should locate inside the lobule, but it is not present in this particular picture. Also, in this layer of loose connective tissue between the lobules, we would have large blood vessels, such as artery and the vein, and some elements of nervous system for innervation of the gland. So here we have the um, neurons of the nerve ganglion. It's the intramural parasympathetic ganglion. It innervates the secretory portions, so mainly parasympathetic. Arch innervates salivary glands, and it means that if sympathetic regulation predominates in some moment of our life, for example, in the moment of stress, where we have more sympathetic action, it means that parasympathetic is not active, and it causes the dry mouth because the salivary glands stop to produce and release saliva. This is how the submandibular salivary glands looks like in a real histological section. Here we see the two kinds of secretory end portions. These are the serows. They are composed of serocytes with basophilic cytoplasm and round cell nuclei, and myoepithelial cells. We see on the cell nuclei of myoepithelial cells, they are flattened, and they are located very, very close to the basal portion of the serocyte. And a second type of secretory portion is the mixed one. It is composed of mucocytes with transparent cytoplasm and flattened nucleus, and serocytes at the periphery. They form the semicrests or semilunions of Januzzi. Here we have secretory portions, mixed and serows, and also small intercalated duct, which looks like the group of cell nuclei. It is very, very small. So here we see the lobule of the gland, and here is the interlobular connective tissue. In this picture, we see here the striated duct. It is composed of elongated columnar cells, which have eosinophilic cytoplasm and striations in the basal part of the cell here. They are also surrounded with myoepithelial cells with flattened nuclei, seen here. These are secretory portions. This one is mixed, and this one is serous. In this picture, we have here striated ducts because the cytoplasm of these cells is more oxyphilic than the cytoplasm of serocytes. We should compare the color to the color of cytoplasm of serocytes. Serocytes are basophilic, and if the cell here looks more eosinophilic, then we should decide that this is the striated duct with eosinophilic cells with basal striations. This is also the striated duct, and here another one, and here also. And this is the intralobular duct, because it has two layers of cuboidal cells. We cannot see the shape of the cells, but we see the shape of nuclei. Nuclei are round, so the shape of the cells should be cuboidal. So this is the intralobular duct, which goes after the striated one. Here we see a large, thick layer of loose connective tissue between the lobules of the gland. And here we see the so-called triad, three different elements. So triad, these elements are duct, artery, and a vein. They also come together. So you should be able to differentiate between them three. How can we differentiate the duct? In this case, we have interlobular duct because it is located between the lobules. Inter means between. So it has 2, 3 or more layers of cells, and cells are cuboidal. So we have here round or maybe oval shape of the nuclei. Now we should look to the vein and to the artery. In the vein and in the artery, we have the flattened endothelial cell nuclei in both cases, here and here. And in the artery, we have elongated, circularly located nuclei of smooth muscle cells. So, to differentiate between artery, vein, and the duct, we should look to the shape of cell nuclei. The ducts have round cell nuclei. This is how the submandibular gland looks like on low magnification. These are lobules, which are separated from each other with layers of loose fibrous connective tissue. Inside each lobule, we have the secretory portions. Some of them have mucocytes, so they look more transparent. So these are mixed. Between them, we have the serols. Also, we see here very dark basophilic and small intercalated duct. Here, for example, the smallest one. Then we have striated ducts, which look more eosinophilic than surrounding serocytes. Here we see how several striated ducts are combined into one larger intralobular duct. They all lead here this way. And somewhere between the lobules, here in loose connective tissue, we should search for interlobular duct, but it is not present in this picture. But it is present in this one because here we have large, thick interlobular connective tissue inside which we have the triad. The artery here with erythrocytes inside, the vein with very thin wall and also erythrocytes here, and the duct. The duct is surrounded with the cuboidal cells with round cell nuclei. Here we have the continuation of the artery because of the cross striation seen here, and these two are continuations of this duct. Also in this connective tissue, along with the blood vessels and ducts, we can have the nerves. This is the nerve. It is somewhat wavy looking, and here also. And this can be the ganglion, intramural ganglion containing cell bodies of neurons. Inside the lobules, here and here, we can find a lot of secretory portions, ducts. These are striated ducts because they are more oxyphilic, and they lead to larger intralobular duct. At high magnification, this group of cell nuclei looks like the intercalated duct. Here also we see the intercalated duct. It looks like that group of small cells with round nuclei, and here also intercalated duct. This is the striated duct because of the color of the cells, which look more basophilic than the serocytes. Also here and here we also have the striated ducts, and here and here. Here we have the triad. This is the duct because of round cell nuclei here. This is also the duct because of round cell nuclei, 2 or 3 layers of cells. This is the artery with concentrically circular smooth muscle cells and erythrocytes inside. This is the vein with flattened endothelium and very thin wall. This is the continuation of this vein. And this is the intramural ganglion, which contains large basophilic cells with large light nuclei with nucleoli. And these are the nerve fibers which look wavy. Here and here also we have the lymphatic vessels. So these elements usually go together. Blood vessels, lymphatic vessels, nerves, and ducts. This picture, we see the striated ducts, which have oxyphilic cytoplasm, and this can be the intralobular because it has two layers of cells. Here striated, also striated duct, and this group of cell nuclei can show us the intercalated duct. Here striated duct and secretory portions. Striated duct, this can be the intercalated duct, which is smaller. And now we go to the parotid gland. Parotid gland has only several secretory portions, so it is more evenly stained in the same color, and most of the elements here are the serosecretory portions, and between them we can find the same four types of ducts. The smallest one is the intercalated duct. It is seen here as the group of cell nuclei with a very well seen lumen in the center. Also here we have these intercalated ducts. Then go striated ducts, which also have very well seen lumen, and the cytoplasm is oxyphilic, but it is not very well seen in such low magnification. Here also the striated duct, here and here and here. After striated ducts go intralobular with two layered epithelium. So this one should be the intralobular, inside the lobule, the largest here. And between the lobules in this connective tissue, we should have interlobular one here. The same at high magnification. These are evenly stained, the serocytes of serosecretory portions. Also here should be the myoepithelial cells with more flattened cell nuclei, such as this one. And here in this picture we also see a couple of intercalated ducts, the smallest ones, which look like a group of cell nuclei and myoepithelial cells. This one is the striated duct with oxyphilic cytoplasm, and here the smaller intercalated duct. This is again the striated duct because of the color of cytoplasm, which is more oxyphilic than the cytoplasm of the serocyte. These are the arterioles because of the concentrically circular located nuclei of smooth muscle cells. This one is the interlobular duct in the parotid gland because it is located inside the thick layer of connective tissue between the lobules. It has two layers of cells which have round cell nuclei. It can have more than two layers, but this one has just two, and no myoepithelial cells. Those cells which have flattened nuclei around the duct are just the cells of loose fibrous connective tissue. We see something inside the duct. This can be the saliva. The saliva in the parotid gland contains a lot of proteins because it's the parotid gland which is composed of protein-producing cells. So it can be slightly eosinophilic in sections because the proteins are usually eosinophilic. Again, we see the parotid gland at low magnification. We see here several lobules separated with interlobular connective tissue, serosecretory portions, small intercalated duct, here again intercalated duct, then striated duct, and intralobular duct. Is located inside the lobule but has rounded two-layered wall. And this is the interlobular duct. Striated duct here, striated duct here, and this one can be the intercalated duct. Here we see small intercalated ducts, and this one should be intralobular duct. It has two layers of cells. It is rather large, but it is located inside the lobule. Now we go to liver and gallbladder. Liver is composed of structural and functional units called hepatic lobules. They have this elongated shape in three-dimensional view, and this is how they look like in horizontal section. If we make the section like this, we will see these hexagonal with 6 angles units called hepatic lobules. Here is one such lobule. Between the lobules, we have the surrounding loose fibrous connective tissue, which contains blood vessels, nerves, and the bile ducts. Inside the center of each lobule, we have the central vein, and the blood flows from the periphery to the central vein through the sinusoidal capillaries. So here we see several lobules separated from each other with layers of loose fibrous connective tissue. And at the center, we have the central vein. Into the central vein, blood flows through the sinusoidal capillaries. Between the plates of the main cells of the liver called hepatocytes. So these are hepatocytes, they form the plates. They are seen as eosinophilic structures here in this picture. The lighter areas between eosinophilic are sinusoidal capillaries. This is how the liver of a pig looks like in the staining called picrofuchsin iron hematoxylin. Picrofuchsin is the staining which stains mainly connective tissue in more intensely orange color. All other structures look like the yellowish background. Iron hematoxylin stains all cell nuclei in grayish or bluish color. So this staining was used to see better the connective tissue which separates lobules from each other. So we have these areas of loose connective tissue which show us the borders of the lobules. So here we see one lobule with the central vein in the center. And this is the second preparation of liver in our class, which is the human liver stained with hematoxylin and eosin. Human liver has much less amount of loose connective tissue, and in normal liver we almost have no connective tissue or very, very few amount of connective tissue between the lobules. So it is very, very hard for us to see the borders between the lobules in the human liver. In this specimen, the border should be somewhere here between two lobules. The center of one lobule is here, the central vein, and the center of second is here, the central vein. So here should be the border between those lobules with loose connective tissue, but amount of connective tissue is very few, as I said previously. In pathological conditions such as fibrosis and cirrhosis of liver, the amount of connective tissue increases largely, and the borders of lobules become very well seen, but not in normal liver. So in normal liver, we should look to the blood vessels to define the borders of the lobules. Also, you can see that here at the border, the hepatocytes look more intensely red, and the color here is darker than more closely to the center. This is because of different functions of hepatocytes located at different levels from the periphery to the center of the lobule. So they have different kind of staining of cytoplasm. And also here at the periphery of the lobules, we have the so-called terminal plates of hepatocytes, where we have the Campbell stem cells for hepatocytes. So the liver, as well as other digestive glands, contain cells of growing type of population. If you remember, we have three types of populations according to their ability to renew themselves. They are called first renewing. This is, for example, for the intestinal epithelium, which should be renewed constantly. Second type is growing, which can renew in case of damage, for example, or trauma. This is true for salivary glands, the liver, and the pancreas, discussing today. And also we have the static or stable type of population, which cannot renew itself because it has no stem cells. So here in liver we have stem cells. They are located in terminal plates here. So this is why this area looks more dark as any other area in any other organ which contains stem cells. It always looks more dark than other components of tissue. Here in this specimen we also have the border of the liver. It is covered with the capsule of dense fibrous connective tissue covered with mesothelium. It is called the Glisson's capsule. It contains also some amount of nerve fibers, including the afferent fibers. So in the case of increasing of liver, the pressure to this capsule increases and it causes the pain feeling of the patient with increased liver. Now, the blood supply of the liver. The very specific characteristic feature of the blood supply of the liver is that the liver receives not only arterial blood through artery, but also the venous blood through the vein. And what is more interesting, that the most volume of the blood which comes into the liver is venous. It comes through the portal vein. About 75% of incoming blood is the venous through the portal vein, and only 25% of incoming blood comes through the hepatic artery. So, why so? The arterial blood brings here oxygen. It is oxygenated, but the venous blood, which comes through the portal vein, brings here the blood which was collected from the intestines, from the gastrointestinal tract. It means that everything that was absorbed in our gastrointestinal tract first goes into the liver. This is because of the need to find some toxins and potentially dangerous molecules and substances, and here in liver there will be the detoxification of the toxins. But this also means that the liver receives the most amount of the nutrients than any other organ, because all the nutrients that were absorbed in intestine come first into the liver. And here in liver they are processed and stored. So the liver is the storage for fats and for proteins and for carbohydrates in the form of glycogen. So the vein and artery come into the liver, and then they go always together, accompanied with the bile ducts. So they always go in these three parts, and they called triad. So triad is one artery, one vein, and the one duct. and they split into smaller and smaller branches, and all these branches also go together and form smaller and smaller triads. And these triads can be seen in our histological preparations in connective tissue between the lobules. They always go together. So here the blood supply is shown in this diagram. So blood comes through hepatic portal vein and hepatic artery, which split into the segmentary veins and arteries, then into interlobular, which means between the lobules, which go in connective tissue between the lobules. Then blood goes into the circumlobular artery and circumlobular vein. Circum means around, so these blood vessels go around the lobules this way. And from these circumlobular vein and circumlobular artery, the blood then goes to the sinusoidal capillary and flows from the periphery to the center, to the central vein. And here in sinusoidal capillaries, the blood becomes first mixed, because every sinusoidal capillary receives blood from both vein and artery. So in sinusoidal capillaries, blood first is mixed, and then when it comes into the central vein, the blood becomes completely venous, because it gave all the oxygen to the hepatocytes while it flew from the periphery to the center. The central vein goes longitudinally through the lobule. So in this picture, it goes downwards this way, and central veins go into sublobular connective veins, which then go to hepatic vein and then to the vena cava. So here, the same in this diagram, blood entry happens through the hepatic portal vein and hepatic artery, which split then into lobar arteries and veins, then segmentary, then interlobular arteries and veins, circumlobular arteries and veins, then into sinusoidal capillaries, where the blood is mixed, and hence this color of this frame. And blood drains through the veins, central vein, then sublobular, collecting, hepatic, and inferior vena cava. In these diagrams, we see the 3D drawings of hepatic lobules with the central vein in the center, which then goes to the sublobular vein. It collects blood coming from the periphery through the sinusoidal capillaries, which go between the trabeculae of hepatocytes. These lighter cells are hepatocytes. They form trabeculae, and between those trabeculae run sinusoidal capillaries. The veins, arteries, and bile ducts come together and form triads. They include interlobular artery, interlobular vein, and interlobular ducts here. These blood vessels can be circumlobular because they go around the lobule. As I said previously, it is very hard for us to see anything in the preparation of human liver because of almost absence of connective tissue normally. So we should look to the blood vessels to see anything on this preparation. So first of all, central vein, how it looks like. It is the vein which lacks muscular tissue, so it is the fibrous vein. It has only endothelium in its structure, and it is single. No other elements is seen here. No artery, no other vein, nor bile duct, nothing. Just the cross section of the very thin walled vein. And also from the central vein, the sinusoidal capillaries and the trabeculae of hepatocytes run equally in all directions. This is called radially oriented trabeculae of hepatocytes and sinusoidal capillaries in all the ways. So if we see such blood vessel, it should be the central vein, and we are in the center of the lobule. Next, if we see 2 or 3 or more elements together, such as artery, vein, bile duct, also here can be also lymphatic blood vessel, or maybe 2 ducts or 2 arteries and vein and bile duct. So 2 or more elements, usually 3, but the number can differ. This is definitely the hepatic triad. It should contain artery, vein, and the duct. So we are now in the connective tissue between the adjacent lobules. Next, if we see again single blood vessel, no other element is seen around, no artery, no vein, no duct, only this vein, but it still has thin wall, but in its wall also muscle cells can be present and a thicker layer of connective tissue. And what is the most important? The direction of this blood vessel is longitudinal, so it is longitudinally sectioned, unlike the central vein, which is cross-sected in our histological preparations. This one is sectioned longitudinally, so we see the elongated section. This should be the sublobular vein. Sublobular vein is the single vein which collects blood from central veins. So let's try to use this on histological preparation. So here in the center, we see the central vein, because we see how elements go equally in all directions, radially. These are sinusoidal capillaries between the trabeculae of hepatocytes, and no other elements we see here around. So this should be the central vein. The same we see here, 1 blood vessels and sinusoidal capillaries and hepatocytes run equally in all directions, radially from it. Now here we see some elements grouped together. It is too small magnification to see how many elements we see here, but definitely more than 1. So this should be the triad, and we are between the lobules here. The same is here in this area, because also we see here more than 2 elements. In this picture, at high magnification, we see the central vein. Into central vein, the sinusoidal capillaries drain. Between sinusoidal capillaries, we have the trabeculae of hepatocytes. Hepatocytes have mostly eosinophilic cytoplasm and round cell nuclei. Some of hepatocytes can have 2 nuclei. It increases the functional activity of these cells. In the sinusoidal capillaries, we have endothelial cells and some other cells which can be present in these capillaries. At the periphery, we have connective tissue which contains blood vessels and bile ducts. So central vein is amuscular. It has very thin wall composed mostly of endothelial cells. Endothelial cells and then hepatocyte starts. So very, very thin wall, and sinusoidal capillaries run in all directions radially. We can easily say where the capillaries here are located because of shape of flattened endothelial cells. The round cell nuclei are the nuclei of hepatocytes. Elongated cell nuclei, mostly of endothelial cells of sinusoidal capillaries. Here we see the scanning electron microscopy picture, and we are kind of inside the sinusoidal capillary. We see its wall from inside. We see that it is really sinusoidal because it has a lot of holes and gaps inside the wall. And the components of blood plasma can easily pass through these holes from the blood capillary to the cells, hepatocytes. In this picture, the specific staining, carmine staining, was used to visualize the blood vessels. So it is again the human liver, so we cannot see here very well developed loose connective tissue, and we should look to the structure of blood vessels. For example, this one. This one is single, no other elements are seen anywhere around this one, and we can say that hepatocytes run in all directions evenly from this. So this can be the central vein. Here we see more than one element, one large and 2, 3, and 5 other round structures with holes in the center, with lumen. So this should be the triad, and we are between the lobules. The same is here, one large and 1, 2, 3 smaller round elements. This is again the triad. The same is here, at least 1, 2 elements together, so it is triad. Here again, they are at some distance from each other, but they are also the elements of triad. And if we look here, we see the longitudinal section of the single blood vessel. It is the sublobular vein. It is the central vein. It is single, very thin wall, and hepatocytes and sinusoidal capillaries run evenly in all directions. Now, the ultrastructure of hepatocytes and the poles of these cells. Hepatocytes are epithelial cells which derive from endoderm. They have the connections between each other, the intercellular junctions, including tight junctions. One surface of the hepatocyte looks to the sinusoidal capillary, and such surface will be called the vascular pole of the hepatocyte. Vascular because it faces the blood capillary. In this picture, here we see the round gaps and holes in endothelium. So this is the wall of the sinusoidal capillary. And here we see the microvilli of the hepatocyte facing this blood capillary. So this should be the vascular pole of the hepatocyte. One hepatocyte can have more than one vascular pole, as it's shown in this picture and in the second one, because the opposite pole also faces the blood capillary. So this also will be the vascular pole of the hepatocyte, and here the same. This is the erythrocyte in blood capillary. And also the hepatocyte has the so-called biliary pole. One of the major functions of the liver and of hepatocytes is to produce bile, which is released into very tiny, very small blood capillaries, biliary capillaries. Here they are located between adjacent hepatocytes, and their walls are made up with cell membrane of the two adjacent hepatocytes. So the bile capillaries are just very tiny spaces between the two cells. Here we see them on this picture. One cell, second cell, and here between them this tiny space, and the microvilli come from one and from the second hepatocyte into this biliary capillary. The same is here. Biliary capillary is here, and biliary bile capillaries are enclosed with tight junctions to ensure that the bile will never come into blood normally. This is how it looks like in the real electron microscopy picture. Here we see number 4 is for the bile capillary between the two adjacent hepatocytes. So these arrows, this one and this one, point to the complex of junctions, including tight junctions, which seal here the two membranes of two adjacent hepatocytes, which make this bile capillary, to ensure that bile will stay here and will not leak between the cells to the sinusoidal blood capillary. So on the left, we see the part of the cytoplasm of first hepatocyte, and on the right of the second one. What do we see here? Number one stays for the part of the nucleus of the first hepatocyte. Here in its cytoplasm, we have some mitochondria. Number two is for mitochondria. These number three are lysosomes. The picture tells us that the lysosomes have the bile pigments. So near this biliary pool, it means the cytoplasm, which is the most closely located to the bile capillary, we will have those organelles which serve for production of bile, because bile will be released into these bile capillaries. The bile contains the biliary pigments, so here we have the lysosomes which process these biliary pigments. Also here we should have a lot of smooth endoplasmic reticulum, which is seen better on this picture. It shows the same, but here we have the high magnification. This is the bile capillary. Its wall are the walls are made with the membrane of the first and of the second hepatocytes. Microvilli are seen here. Arrows point to the intercellular junctions between the hepatocytes, and we see what is well developed in the cytoplasm of these cells. Here we have a lot of smooth endoplasmic reticulum, which looks like the vesicles and tubules and cisternae of different shape. These are the major organelles which serve to produce most of the components of the bile. The bile contains the phospholipids, some other lipids, cholesterol molecules, so they all are produced by smooth endoplasmic reticulum. So this is why it is very well developed in this part of the cell. Here the metabolic and secretory processes in hepatocytes are shown. In the left picture, the metabolism of glucose is shown. So the hepatocyte takes the glucose from blood. The hormone insulin should act upon the hepatocyte to ensure the intake of glucose. Glucose then goes to smooth endoplasmic reticulum, which turns the glucose into the polymer molecule called glycogen. And glycogen is stored in granules seen in black color. So these black are granules of glycogen. The liver is the main organ for storage of carbohydrates in the form of glycogen. When it is needed, when the signal from the hormone glucagon comes to the hepatocyte, it can break down the glycogen and release glucose into the bloodstream. Also, the smooth endoplasmic reticulum serves for the metabolism of lipids, because the lipids can be stored and produced in smooth endoplasmic reticulum, and liver also can store the lipids in form of lipid droplets produced by smooth endoplasmic reticulum. And also, smooth endoplasmic reticulum is the organelle which provides detoxification of toxins. As I said previously, all blood from intestines comes to liver for detoxification of something potentially dangerous from our food. And this detoxification happens also in smooth endoplasmic reticulum. What about the proteins? The proteins come into the hepatocyte in the form of amino acids, because amino acids come with blood from intestines, and amino acids come into rough endoplasmic reticulum where the proteins are synthesized. The liver synthesizes a lot of proteins. Among the most important are the proteins of the blood plasma, such as albumin, fibrinogen, prothrombin, and lipoproteins. So these proteins of blood plasma are produced in rough endoplasmic reticulum in hepatocytes and released via exocytosis. Here in the right picture, we have the recycling of the bile acids. Bile acids is one of the components of the bile secreted through the bile capillary by the hepatocyte. And these bile acids are produced also in smooth endoplasmic reticulum. So these bile acids are produced when the cholic acid is conjugated with glycine and taurine here, and these bile acids are released into the bile capillary, go to the duodenum, where bile goes through the ducts. Then in duodenum, bile helps digestion process, especially digestion of lipids, and some amount of these bile acids is absorbed by the enterocytes with brush border inside the small intestine. What is absorbed goes into blood, and with the bloodstream, these bile acids can come again into the liver. They are now reabsorbed in the gut, come with blood into liver, and go again into the bile capillaries. So about 90% of bile acids are recycling this way, from liver to intestine and with blood back again. And 10% are newly synthesized in smooth endoplasmic reticulum. Here we see the central vein and hepatocytes stained with PAS reaction. This PAS staining, PAS reaction, is for visualization of glycogens, to see glycogens. Glycogen and other sugars. So the glycogen is the large molecule for storing the glucose in our body. So these small pink dots found in cytoplasm of the hepatocytes are the glycogen molecules. As you see here, some hepatocytes have two nuclei. Some of them can have one but larger nucleus, larger than other. In both cases, in one but larger or too small. This is the polyploidy of the hepatocytes. It serves for increasing the intensity of functions of these cells. So bile produced by hepatocytes should go into the duodenum. It contains water, ions, bile acids, bile pigments, cholesterol, and phospholipids. There are no enzymes in bile. Water, ions, bile acids. We have discussed previously about bile acids. Bile acids serve for emulsification and helping to digest lipids. Bile pigments are a result of the processing of dead erythrocytes. Erythrocytes are destructed in spleen, and some of them are destructed in the liver, and the hemoglobin is destroyed by the macrophages, Kupffer cells of the liver, and are processed then by hepatocytes into the bile pigments. And these bile pigments should go with the bile to the intestine. Some of the bile pigments go to intestine and give the feces the brown color, and some of the bile pigments go to the blood and then into the kidney and to the urine, making the urine its yellow color. So bile pigments are just the result of destroying of hemoglobin, because the hemoglobin and its metabolism products can be toxic for the body. So they should be transformed into non-toxic forms by the hepatocytes and released from the body with urine and the feces. So bile pigments in bile are just the waste products. Then cholesterol and phospholipids in bile also help to provide the digestion of lipids. No enzymes we have in bile. Enzymes come only from pancreas. Functions of bile. First, it stimulates secretion of pancreatic and duodenal juice. When it comes into the duodenum, it acts upon the cells, endocrine cells, which release their hormones, which activate the production and release of pancreatic juice and the duodenal juice. The secretion of the exocrine pancreatic glands and secretion of duodenal Brunner's glands. Second, bile stimulates intestinal enterocytes proliferation and desquamation. Next, bile activates pancreatic and intestinal enzymes. It provides the needed pH level and the environment for these enzymes, for their activation. As I said previously, it helps digestion of lipids. It helps emulsification process of lipids. Emulsification means the physical and mechanical break of larger droplets of lipids to smaller ones, because the enzymes, lipases, which come from pancreas, should then attach to these droplets of lipids from the outside and start to break down these molecules. So as smaller the droplets are, as faster the process of the breaking and digestion of these droplets will happen. Next, the bile promotes absorption and products of lipids, hydrolysis, and fat-soluble vitamins in the intestines. Then it provides resynthesis of triglycerides in enterocytes. Next, it inactivates pepsins and also neutralizes the acidic chyme coming from the stomach, because it has a little alkaline pH level. Next, it enhances intestinal motility, and it has a bacteriostatic effect on intestinal microorganisms. This is for the task in your album. In some of the tasks, you should mark correctly the bile flow. So, bile is produced and released by hepatocytes into the bile canaliculus or bile capillary. So first it flows through the bile canaliculus from the central portion of the lobule to the periphery. So at first the bile canaliculus wall is just the membranes of adjacent hepatocytes. Near the periphery of the hepatic lobule, there is the canal of Herring. It has in its structure some portions of membranes of hepatocytes and specific cells, cuboidal cells of the first portions of the bile ducts. Then goes cholangiole. Cholangiole is actually the circumlobular duct. It goes around the lobule, as well as circumlobular artery and the vein. So they go in three and make this circumlobular triad. And after the circumlobular cholangiole, there will be the interlobular bile duct. Interlobular bile duct that along with the artery and the vein, also called interlobular, make the interlobular triad. So in this picture, we see the central vein in the center of the lobule. These are the hepatocytes making the plates. So these are the plates of hepatocytes. Between the plates of hepatocytes, there is the bile capillary or bile canaliculus. This arrow shows the way of the bile flow. So it flows from the center, always between the hepatocytes, to the periphery, then into the canal of Herring. It is better shown in this portion. It starts to have its own cells in its structure. Then goes the cholangiole. It is the circumlobular bile duct. It goes along with circumlobular vein and artery around the lobule this way. And from the circumlobular duct cholangiole, there will be the interlobular bile duct, which along with interlobular artery and interlobular vein make the hepatic or portal triad. Also in this area we can find the lymphatic vessel and some nerve elements, as was previously in the salivary gland. In this electron microscopy picture, we see the interlobular bile duct, which is composed of one layer of cuboidal cells called cholangiocytes. Cholangiocytes, epithelial cells derived from endoderm, as everything in liver, the parenchyma. The cells are interconnected with the number of intercellular junctions, including the tight junctions near the apical surface. Then belt desmosomes and desmosomes, etc. Here is the basal membrane. In this lumen, there is the bile. So in this picture, again, we see the hepatic lobules. We see the center, the central vein. In interlobular connective tissue, we have the portal or hepatic triad, which contains larger element, the vein, and smaller ones, artery and ducts. So in hepatic or portal triad, we will have 2 or 3 or more elements. The largest of them is the vein. As I said previously, about 75% of blood which comes into the liver is the venous in vein. So the veins are larger than arteries, and so the largest element always will be the vein. And others should be the artery and bile duct. How to differentiate between them? We should look to the cell nuclei. In bile duct, we have lighter stained oval or round in shape nuclei. In artery, we have endothelial cells inside and elongated cell nuclei of smooth muscle cells, concentrically located, circumferential. We can have here also smaller other elements like here and here and here and here. They are smaller branches of these larger vein, artery, and the duct. In this specimen, also we have the triad. This one should be the artery because of these concentrically located smooth muscle cells. This is the duct because of the shape of the nuclei, which are round or oval in shape. And this is the vein with the largest lumen and the thin wall. Also here we see the smaller elements like this one. This is again the duct because of the shape of the nuclei, which are round to oval, and here also. So we are here between the 3 adjacent lobules of the liver. One lobule is here in the upper left corner, second is here on the right, and third is here downwards. So here we see the area where the hepatic globules have stem cells for hepatocytes. So here the volume of cells is smaller than closely to the center of the lobule here. This is because here we have the stem cells, which have usually smaller cytoplasm. Another example of the hepatic triad, the largest element is always the vein, with its flattened endothelial cells here. This should be the artery because of elongated, circumferentially located smooth muscle cells, and this one, the duct, because of the oval shape of the nuclei. This one, smaller artery, again smaller artery, here duct because of round cell nuclei, here also duct. And now we go to the structure of sinusoidal capillaries and the cells located there. In this picture, which is the same picture that you have in the task in your album, we have some hepatocytes connecting together to form the plate of hepatocytes. Between the plates of hepatocytes, we have the sinusoidal capillary. So hepatocytes have the vascular pole, which faces the capillary. This hepatocyte has three vascular poles. This one, because here also capillary, and this one, because here also capillary, blood capillary. And biliary pole. Biliary pole is the cytoplasm which faces the bile capillary or bile canaliculus. Bile canaliculi are enclosed between adjacent hepatocytes inside the plate of hepatocytes. Now, we should look to the structure of the sinusoidal capillary. Its wall is made up with the endothelial cells, which lack the basement membrane and have a lot of gaps and holes in their cytoplasm. Also, we can have here the Kupffer cells, the specific macrophages of the liver, which can perform the function of phagocytosing of any toxins, the immune complexes, the damaged and dying erythrocytes, bacteria, etc. So they perform the defensive function. As you can see here, there is the space between the endothelium and hepatocyte. Here is this space where the microvilli of hepatocytes are located, and this space is called the perisinusoidal space of Disse. In this perisinusoidal space of Disse, we have the blood plasma, which can easily pass through these gaps and holes in endothelium, and here the processes of exchange between the blood and hepatocytes happen. So hepatocytes release here produced proteins of blood plasma, such as albumin and fibrinogen, and take from blood plasma everything that they want to, such as glucose, amino acids, toxins, etc. So near the vascular pole, there will be a lot of organelles, such as rough endoplasmic reticulum. It should be located here, because here from the vascular pole, the plasma proteins should be released in blood plasma. Also here near the vascular pole, the smooth endoplasmic reticulum is developed for production of glycogen coming from blood plasma, glucose, production of lipids, and also detoxification of toxins coming from blood. Also, if we look to the space of Disse, we have here these worm-like elongated structures, which are the reticular fibers. Reticular fibers are made by these triangular cells called perisinusoidal lipocytes or Ito cells. These perisinusoidal lipocytes, as comes from their name, serve for production and storage of lipids in the form of these lipid droplets in their cytoplasm. And their second function is to produce the reticular fibers. It happens in normal condition. In pathology, these Ito cells, perisinusoidal lipocytes, become activated and start to produce a lot of collagen fibers, which start to fill this DISC space. And then if we have the collagen fibers here, it causes interruption of exchange between blood plasma and hepatocyte, and it will cause the death of hepatocyte. So this process will be called fibrosis and cirrhosis. So this is the function of Ito cells. Also, in this picture, we have another cell type not shown in the previous one, so we use here two pictures. Here we have other cell called PIT cell. PIT cell is actually the natural killer lymphocyte, so it has round cell nucleus, small amount of cytoplasm and pseudopodia, and it performs, along with the Kupffer macrophage cell, the function of defense. As any natural killer lymphocyte, it acts like the T killer. It kills the cells that are not normal. So it can kill the ill or not normal hepatocytes, which do not produce the correct proteins upon their MHC1 proteins upon their surface, or the cells which were infected with viruses, etc. Also, it releases the granules, which activate the macrophage and other cells here. So it acts in local reactions. In this picture, we see the Ito cell, which stores the lipid in lipid droplets and also the lipid-soluble vitamins. Here in this slide, there are all the cells with their functions. You should learn this information. In this picture, we have the reticular fibers, which surround sinusoidal blood capillaries. This histological preparation was stained with silver salts. Silver salts stain reticular fibers, and so reticular fibers are called argyrophilic because of this. So here is the sinusoidal capillary, and reticular fibers wrap around the sinusoidal capillary, giving its strength, some strength, because sinusoidal capillary are very thin, they have no basement membrane, and they cannot maintain their shape. In this picture, the ink of black color was added to blood of the laboratory animal, which section of the liver we see here, and we see that all the cells that are capable for endocytosis have endocytosed the ink from blood. Such cells here are the Kupffer cells, macrophages, so we have that their cytoplasm now is filled with black granules of this black dye, black ink. The same on high magnification, we see here these Kupffer cells, macrophages, that reside in sinusoidal capillaries and perform here the function of endocytosis. So they have here the nucleus and in cytoplasm granules of black ink. This is the electron microscopy picture of the sinusoidal capillary of the liver. These are red blood cells in its lumen. This is the portions of the endothelium, because it has large gaps and pores in its membrane. Here is the space of DISC between the endothelium and hepatocytes. Hepatocytes have microvilli upon their vascular pole. So here is the vascular pole of the hepatocyte, the nucleus of hepatocyte, mitochondria, rough endoplasmic reticulum here, smooth endoplasmic reticulum. The functions of liver are shown here. You should learn not only the functions of liver, but you should be able to explain which cells of liver perform these functions. So, metabolic function of liver includes participation in metabolism of carbohydrates, proteins, lipids, pigments, vitamins, mineral components, etc., including the synthesis of glycogen, phospholipids, cholesterol, formation of blood plasma proteins, for example, albumin, fibrinogen, and prothrombin. So, most of these functions are produced by the hepatocytes themselves. Next, secretory function, exocrine function, the formation of the bile and its release into the duodenum, and endocrine function is the release into the blood of the synthesized products. So it's again about the hepatocyte. Next, deposition, accumulation and storage of blood, vitamins, A, D, E, K, which are some of them lipid soluble, some of them water soluble, storage of glycogen and microelements. So most of these functions are also produced by hepatocytes, but some of them are produced by other cells, such as Ito cell, which stores the lipids and lipid-soluble vitamins. Waste removal. Products of worn erythrocytes, such as bile pigments biliverdin and bilirubin, urea, worn hormones are removed from liver. So mostly it's again the function of hepatocytes, but in the processing of erythrocytes, the Kupffer cell also participates, so it's the function of Kupffer macrophages. Also, endocrine function, the production of hormones such as hepcidin, somatomedin, etc. It's again about the hepatocytes. Function of defense, the activation and detoxification of toxins and xenobiotics. Xenobiotics means some organic molecules that come from outside, that are not characteristic for our organism. This happens in hepatocytes, in particularly in smooth endoplasmic reticulum. Removal of the worn erythrocytes. This is the combined function of macrophages and hepatocytes. Destruction of bacteria and other antigens. It's about the macrophages, Kupffer cells. Immune reactions. It's about the macrophages and also the PIT cells. And also destruction of tumor cells and virus-infected cells. It's about the PIT cells, which destroy such bad cells and cause the apoptosis in these cells. And then when apoptosis happens, the dead cell is eaten with the macrophage. And finally, the hematopoietic function of liver. As you remember, the formation of second generation of blood stem cells and blood cells happens from the 2nd to the 8th month of the embryo life in liver. It's the second period of embryonic hematopoietic poiesis. So it is called hepatolional period because it happens mainly in liver and to some extent in spleen. And in some pathological conditions, for example, in the case of failure of red bone marrow for some reasons, the hematopoiesis can happen again in liver, as it was previously during embryogenesis. So liver is capable for hematopoiesis. We discussed previously the classical lobule, which is hexagonal with 6 angles. It describes the blood flowing inside this lobule from the periphery to the center. Also, we can discuss the structure of liver in different terms. For example, in the term of the portal lobule, it will be triangular in shape with each angle inside the central vein of the lobules. And this portal lobule describe the bile flow. So the bile will flow from the periphery of this portal lobule to the center, especially to the bile duct, particularly to the bile duct inside the portal triad. So this portal lobule is usually described when discussing the disturbances of the bile drainage inside the liver. And also we can use the term hepatic acinus, which is the rhombus in shape with angles in central veins and in portal triads. It describes the blood supply of different hepatocytes. It is better shown in this picture. So here is this acinus shown. So the hepatocytes can be subdivided into 3 zones: zone 1, 2, and 3. As you remember, the blood flows from the periphery of our classical lobule to the central vein, from the periphery to the central vein, like this. And so the blood will flow from these circumlobular blood vessels to sinusoidal capillaries, this way. So blood first will come to the hepatocytes of zone 1, then to hepatocytes of zone 2, and only then to hepatocytes of zone 3. It means that while the blood flows through these zones, first oxygen is taken by the hepatocyte of zone 1, then the blood comes to the hepatocytes of zone 2, but the amount of oxygen is now lower because it was partly taken by the previously hepatocytes here in zone 1. And the blood which comes to the hepatocytes of zone 3 is very poor of oxygen. And it means that the hepatocytes of the zone 3 are the most vulnerable during hypoxia. It means that if the blood brings to the liver a very small amount of oxygen for some reasons, all the oxygen will be consumed by the hepatocyte of zone 1. And the rest of oxygen will be taken by the hepatocytes of zone 2, and no more oxygen will come with blood to hepatocytes of zone 3. So they will die first in the case of hypoxia. Opposite, the hepatocytes of zone 1 will be more vulnerable to toxins because they are first who will meet the toxins if they come from blood, from intestines. So they will receive a larger amount of the toxin and they would die first. Some more pictures of our histological preparations. This is the liver of a pig stained with picrofuchsin iron hematoxylin. So here we have yellowish brownish background with the violet cell nuclei, and connective tissue is more intensely stained for us to be ready to see the borders between the lobules. Pigs, unlike humans, normally have larger amounts of loose connective tissue between the lobules. So we use the preparation of a liver of a pig for students to help them to see the borders between the lobules better than in human liver preparations. So here we see the triad. The largest element of the triad is the vein, portal vein. It is filled with erythrocytes. Erythrocytes here have yellow color. This is the second element. This should be the bile duct. And here is the third element. Maybe it is the artery, but we cannot say it because of low magnification. Here we have the central vein filled with yellow erythrocytes. Here we see the borders between the several lobules. This is the triad because here we see the vein filled with erythrocytes and two other elements. We cannot say exactly where is which. This is the longitudinal section of some single vein. So this is the sublobular vein. And here should be the central vein. It is oblique sected, so some of the section came here. This is better picture because here we have the classical shape of the lobule and the central vein in the center and several triads seen at the periphery, with the largest element, the vein, filled with erythrocytes. And back to the human liver, now we know how to differentiate the elements here. So this is the central vein because it is the single, and the sinusoidal capillaries and plates of hepatocytes run equally radially in all directions. The same we can say about this blood vessel. So these are the centers of the adjacent classical lobules. Here is the triad because here we have large vein and something here, maybe duct, maybe artery. And here also we have one object here and other here at the periphery. So this is again the triad, and we are between the lobules. Another example of the human liver preparation. This should be the central vein. It is single and elements run equally in all directions. This is the triad because here we have the vein and here smaller other elements, ducts and arteries. The same is about this. This is again the triad. The same about this large vein and something smaller here. And this is the sublobular vein. It is single but longitudinally sected. This is the central vein, a little bit oblique sected, but we can see that the hepatocytes run equally in all directions from it. This is the border between the lobules, and here we see the triad, this area, and here also, because here we see several elements. This is again the central vein, because the hepatocytes run equally in all directions. At high magnification, here in the lower part of the picture, we see a very small triad. It is composed of very small bile duct, very small artery or maybe arteriole, and the small vein or a venule. As I said previously, veins, arteries, and bile ducts come together and branch together into smaller, and smaller branches. And here we have the smallest branch of them three, but still they come together and form the triad. These are hepatocytes. Between hepatocytes, we see these spaces. Inside these spaces, we see the cell nuclei, which are elongated. These are cell nuclei of endothelial cells. So here we have sinusoidal capillaries. Also in sinusoidal capillaries, here we see the small round cell nuclei, which resemble cell nuclei of lymphocytes, and these can actually be lymphocytes. I mean PET cells, which are natural killer lymphocytes. Also, here this can be the Ito cell with triangular nucleus, and here also. And macrophage can have such nucleus, which is the bean shaped. Other nuclei which are round and light in color with the nucleolus in the center, those are nuclei of hepatocytes. Another example of the triad, this is the duct. because of the shape of the nuclei here. This can be the artery, but very small, and this should be the vein, because it is the largest element here. Now we go to the gallbladder. It is the thin-walled sac which serves for storage and concentration of the bile produced by liver. So gallbladder just stores the bile but doesn't produce any components of this bile. It is needed because the liver produces bile constantly, but the bile itself should come to the duodenum through the system of the ducts, and it is needed for digestion process. And because bile is produced constantly, but we do not eat constantly, so between the meals, the bile should be stored somewhere, and it is stored in the gallbladder, and it is released through the ducts into the duodenum when the food comes into our mouth and into our stomach. There, the cells feel that we have eat something, and they send the signals to the gallbladder, and it contracts and releases the bile. As you see here, the biliary system has the same duct with the pancreas. And because of the shape of the gallbladder and because of the concentration of bile, it can happen that the stones, bile stones, can start to be formed in the gallbladder. And if the stones come through the ducts this way, they can stop here because the opening of the duct has the sphincter. It is not very large, so the stone can stuck here in this position, and it will not let the bile to come through the duct into duodenum, and it will also interrupt the coming of enzymes from pancreas into the duodenum, and it can cause the necrosis processes in the pancreas, because there will be the enzymes which are able to digest any organic, including organic materials of the cells of the pancreas in itself. So the gallbladder wall is rather thin. The functions of gallbladder are to store the bile, to concentrate the bile, and to release the bile when it is needed. So for storage of the bile, so first, in the wall of the gallbladder, we have three layers: mucosa, muscularis, and serosa or adventitia. Serosa covers the gallbladder in those areas which face the abdominal cavity. Here, this surface will be covered with mesothelium, and this surface which faces the surface of the liver, because the gallbladder is located just below the liver. This surface will be covered with adventitia. So in the mucosa, we have just two sublayers: the epithelium, which is simple columnar absorptive epithelium, like in the small intestine with brush border, and lamina propria mucosa. In muscularis, we have two layers of smooth muscle cells, not very well seen. The border between those two layers. And outer layer, as I said previously, serosa or adventitia. So for storage of bile, the wall of gallbladder has the ability to stretch because of the presence of smooth muscle cells. For concentration of bile, concentration means that we should take water from this fluid, and this means the concentration. So to concentrate the bile, we should take the water from it, And this is what the wall of the gallbladder does. This is why it has the simple columnar absorptive epithelium for absorption of water. So water comes from the bile to the cells and then to blood capillaries in lamina propria mucosa. And this is why the inner surface is not flat, but it forms such folds that can be connected together and form these arcades. Arcade means these two connected folds, like the bridge between the two folds. This is the characteristic feature of the gallbladder mucosa. So these folds increase the surface of the epithelium for its function, the absorption of water and concentration of the bile. So as a result of the concentration of the bile which comes directly from liver through the hepatic duct into duodenum is more light and watery. The bile which comes from gallbladder through its cystic duct into the duodenum will be more dark and viscous. And finally, a release of the bile when it is needed. It is performed with the help of the contraction of smooth muscle cells in its muscularis. In this picture, we see the wall of the gallbladder here with its specific arcades, bridging folds of the mucosa. And this is the section of the liver. So here we see the area where the gallbladder contacts with the surface of the liver, and they have common adventitia. So they both are covered with loose fibrous connective tissue, which is common for them both. The same at high magnification. We see here mucosa, muscularis, and adventitia. There is no very sharp border between those layers. We can differentiate the muscularis by the shape of the smooth muscle cells, which have more elongated cell nuclei than the nuclei of surrounding connective tissue. In mucosa, we have the epithelium and lamina propria mucosa. Mucosa forms these arcades, bridging folds. Again, these bridging folds between the columnar epithelial cells with microvilli, there can be found some goblet cells, especially closer to the area where the duct starts. Here we see the highest magnification of this epithelium. The apical surface is covered with microvilli, and what the cells are doing, they absorb water from the bile. How they do this? They have a lot of mitochondria in their cytoplasm. This is why the cytoplasm is eosinophilic. And upon their basal surface, they will have the basal labyrinth with the folds of membrane with a lot of ion pumps. And when the cell wants to absorb water, it should first pump the sodium ions from the fluid. So the cells pump sodium ions from the bile. They take those ions and pump them outside of the cells into connective tissue, and then those ions go to blood capillaries. And the water comes after ions. Water comes through the cytoplasm of the cell, also into connective tissue and into the blood capillaries. So in order to absorb the water, the cell needs first to pump the sodium from the fluid, and then water will come after the sodium. And finally, we'll go to the pancreas. Pancreas anatomically has the subdivisions: the head, which is closely located to the duct system and to the duodenum, the body, and the tail. It is composed of lobules seen from outside. They are separated with loose fibrous connective tissue, and it is well seen on histological sections. So here, like in the salivary glands, we have the lobules separated with loose connective tissue, which form septi. Pancreas has two different portions, the exocrine and endocrine portions, which perform different functions. Exocrine portion is composed of the pancreatic acini, which is the exocrine component of this exocrine gland, and endocrine portion includes the islets of Langerhans. This is the group of endocrine cells in the pancreas. So, two parts, endocrine part and exocrine part and endocrine part. Exocrine part is the compound tubular alveolar branched gland, which includes secretory portions called acini and system of ducts. Endocrine portion is composed of insulocytes, the cells of the islets of Langerhans, endocrine cells. They have no ducts because they should release their hormones directly to the blood capillaries of fenestrated type. This is how the pancreas looks like in the histological sections. These are the acini, secretory portions of the exocrine part of the gland. And this much lighter stained area, this is the islet of Langerhans. As you see here in the islet of Langerhans, the cells have oval-shaped basophilic nuclei and non-stained cytoplasm. So cytoplasm stays pale or non-stained. So the islet looks whitish among a very intensely stained secretory cells of the exocrine portion of the pancreas. This is the connective tissue between the adjacent lobules, which has in its structure artery and the vein. Exocrine part has the acinus, which contains acinar cells. These cells, they produce the enzymes in non-active form. The enzymes in non-active form are called zymogen. So the cells will have zymogen granules in their apical portion, and because zymogens are proteins themselves, the cells needs a rough endoplasmic reticulum and Golgi apparatus. So around the cell nucleus, the cells will have rough endoplasmic reticulum and Golgi complex. This is the ultrastructure of the acinar cells. Also, here we have the centroacinar cells. They are actually the cells of the first type of the duct. It is called intercalated duct because it is kind of inserted inside the secretory portion. So the first cells of this duct are located just inside the secretory portions. They are called centroacinar cells. They are smaller with lighter cytoplasm because they do not produce the enzymes. And from the secretory portion, the ducts start. First one will be called intercalated, then goes in interacinar duct, and then intralobular and interlobular duct. Endocrine portion has only the endocrine cells and blood capillaries, no ducts. In this picture, we see the exocrine part of the pancreas with the acinar cells forming the acinus. Here should be their basement membrane. They have the secretory granules of the zymogen in their apical surface. Intercalated duct, the first duct, which is the smallest, kind of inserted into the acinus, and its cells are called centroacinar cells because they are located inside the acinus, which is shown also in this picture. Intercalated duct is composed of flattened cells with flattened cell nuclei. It is very, very small. So the acinar cell of the exocrine part of the pancreas has two zones. The lower zones, which is called the homogeneous zone, it is basophilic part because it has a lot of rough endoplasmic reticulum around the nucleus and below the nucleus. The apical portion of the same cell is oxyphilic in color. It is called zymogen zone because it contains zymogen granules, granules which contain enzymes in non-active form. The function of acinar cell is to produce the enzymes in inactive form, such as trypsin, chymotrypsin. They are enzymes for proteins digestion, lipase for lipids digestion, amylase maltase for carbohydrates digestion, nucleases for DNA and RNA digestion. Now, how to differentiate between the three similar looking glands? The pancreas and two kinds of salivary glands, submandibular and parotid glands. We should look to the single cell. So, in pancreas, one single cell, acinar cell, has two zones, oxyphilic and basophilic zone. In submandibular gland, in secretory portion, we have two different cells, basophilic cell, serocyte, and pale-looking cell, mucocyte. Also here we have myoepithelial cells. Myoepithelial cells are absent in pancreas because pancreas is the gland derived from endoderm, and such glands derived from endoderm lack myoepithelial cells. Myoepithelial cells are present in glands derived from ectoderm. So, in both cases, we have two colors in secretory portion, but in pancreas, two colors are in the same cell. In submandibular secretory portion, we have two different cells with different colors, serocyte and mucocyte. In parotid gland, all secretory cells are of the same color, because here we have only one secretory cell type, which is serocyte. It is equally stained basophilic, and also myoepithelial cells are present here. In this picture, we see the electron microscopy picture of the acinus and the beginning of the intercalated duct of the pancreas. So this is the acinus. A white area shows the location of the loose connective tissue with blood capillary around the acinus. These are acinar cells or acinocytes. They have rough endoplasmic reticulum around the cell nucleus and zymogen granules in the apical portion of the cells. These zymogen granules should be released here into the lumen of the acinus. Here in the acinus starts the intercalated duct. It starts with centroacinar cells. Another example of the acinus of the pancreas, here we see one such acinar cell. It has a lot of rough endoplasmic reticulum and granules filled with proteins. This is why they are dark stained. They are granules with zymogens. This is the lumen of the acinus. Here we see the connection of the three cells, one cell here, second here, and third here, and the areas of connections between those cells. In the center, this is the lumen of the acinus. And you see that the third and second cells look the same. They have a lot of rough endoplasmic reticulum and zymogen granules. So these two are acinar cells. The first cell has no such developed rough endoplasmic reticulum and no zymogen granules. So this is the centroacinar cell. It is smaller and pale-looking cell. In this picture, the zymogen granules are stained in red color, so they are located more closely to the center of the acinus, and to the periphery of the acinus there is more basophilic staining. Here we have the islet of Langerhans, where the cells have no such granules and the cytoplasm is almost non-stained, very pale looking. The duct system of the exocrine part of the pancreas include 4 types of ducts, the same as in the salivary glands. The first type, intercalated duct. The name is the same as in the salivary gland, but the structure is not the same. Intercalated duct includes squamous simple epithelium with squamous flattened cells with flattened nuclei. No myoepithelial cells, because this gland derives from endoderm. The function of the intercalated duct is to release bicarbonate ions and to release inhibitors of zymogens. So, bicarbonate ions serve to increase the pH level of the pancreatic juice. It is needed to ensure the optimum for the working of the enzymes, because the enzymes of pancreas need more alkaline pH level. Inhibitors of zymogens are the substances that do not let zymogens in active form of enzymes to become active form until they reach the duodenum. Because if the inactive enzymes become active inside the pancreas itself, they would start to digest the pancreatic cells, and it will cause the pancreanecrosis. Next, interacinar ducts. Interacinar means between the acini, between the secretory portions. It is composed of simple cuboidal epithelium without myoepithelial cells, and the cells of interacinar ducts also release inhibitors of zymogens. Next we'll go intralobular ducts and interlobular ducts. They are composed of simple cuboidal epithelium, intralobular ducts, and the largest ducts, interlobular ducts, are composed of simple columnar epithelium. It means that when we go from the secretory portions to the duct leading to duodenum, only the heights, the tall of the cells increases. So at first the cells are flattened in intercalated duct, then the cells become cuboidal in interacinar and intralobular duct, and finally the cells become columnar in interlobular ducts. So only the heights of the cells increases, not the number of layers. The number of layers is always 1, because all the glands which derive from endoderm have always only 1 layer of cells. They are always simple, without any myoepithelial cells. Here in this picture, we see the pancreatic acinus, central acinar cells, and intercalated duct. And also here we see the islet of Langerhans, because it contains cells with non-stained cytoplasm. Here we see the acini, secretory portions of the exocrine part of the pancreas. For example, this is one acinus here. Here is its lumen, and right in the center of the lumen we see the cell nuclei of other cells. These are the cell nuclei of centroacinar cells, because they are called so because they are located just in the center, in the lumen of the acinus. These are acinar cells. Each of them has basophilic homogeneous zone and oxyphilic zymogenous zone. After acini, after secretory portions, ducts start. They start with the intercalated ducts. Here we see the intercalated duct. It is composed of simple squamous epithelium, because here we see the squamous cell nuclei of flattened epithelial cells. In this picture, we see the intercalated and interacinar ducts of the pancreas. This is the interacinar duct. It is composed of cuboidal cells because of round cell nuclei and a very thin layer of connective tissue or almost no connective tissue around it. It is just located between the secretory portions. And here we see the intercalated duct, which is the smallest, and it is composed of mainly flattened cells. Here we see the intralobular ducts, here and here also. They both contain one layer of cuboidal cells, but they are covered with thick layer of connective tissue, unlike the interacinar ducts. But still they are inside the lobules, so they are called intralobular, because they are located inside the lobule. And this one should be the interlobular duct, because it is located in thick layer of connective tissue between the lobules, and it still has one layer of cells. It comprises simple epithelium. Also here you see the acinus with acinar cells. Their nuclei are seen here, and centroacinar cells. Their nuclei are located just in the center, in the lumen of this acinus. These flattened nuclei are not myoepithelial cells, they are cells of connective tissue which surrounds acini. Here we see a very large, or maybe the major interlobular duct of the pancreas. It still has a simple epithelium, but here the cells are columnar in shape. So the thickness of epithelium increases, but still we have only one layer of cells. It is surrounded with loose fibrous connective tissue, very thick layer of connective tissue between the lobules. Inside the tissue of the pancreas, we can find the lamellar corpuscles, Vater Pacini corpuscles, which serve for sense of pressure. If the pressure happens, then it will send the signal to the acinar cells to produce more enzymes. Now, pancreatic islet includes 5 major cell types, such as A cells, which are located mostly at the periphery. Here in the second picture, the immunohistochemistry method was used to show only A cells. Only A cells are stained in more dark color. Other cells are not stained. This is the background staining. So A cells, they produce glucagon. They are located at the periphery. Most of the cells of the pancreatic islet, about 70%, tissue with one major duct lined with simple columnar epithelium. Between the lobules here we have the islet of Langerhans here. This is the interacinar duct because it is very small. It is composed of simple cuboidal epithelium and it is not covered with rather thick layer of connective tissue. It is located just between the acini. Here we see the intercalated duct. It is branching and it is composed of flattened cell nuclei. Another islet of Langerhans here, and maybe here, the tiny one. This can be the blood vessel, and here also blood vessel, the small venule. In this picture here we see the intercalated duct because it is composed of simple squamous epithelium here and here also. In this picture, in this area, we have the islet of Langerhans with non-stained cells, and here some ducts. Here should be the intercalated duct with flattened cells, and here interacinar duct with more round cell nuclei with cuboidal epithelium. At high magnification, here we see on the right the islet of Langerhans with insulocytes and blood capillary of fenestrated type. Here, the flattened cell nuclei of intercalated duct. It is branching this way and this way. Here at high magnification, we see the islet of Langerhans. So that's all. Thank you for your attention and see you in class.